Hi girls, this is the first of 3 videos on free energy and matter. This video will focus on energy, the second on photosynthesis, and the third on cellular respiration. Now, in order to fully understand photosynthesis and cellular respiration, we have to understand the energy flow that's driving these reactions. Where is that energy coming from? Why is it necessary? And how does it help organisms to live? Bioenergetics is the term that we can use when we're talking about the flow of energy in general. Now, when we focus in on a specific organism or cell, we have something called metabolism. Metabolism is the sum of all the chemical reactions occurring within that cell or organism that allow it to live. Metabolism is then made up by several metabolic pathways. Our metabolic pathways occur within the cell, and they allow that cell to function, to grow, and to reproduce. Examples of metabolic pathways are glycolysis and the Krebs cycle and oxidative phosphorylation, etc. Our metabolic pathways are then categorized into two things, either anabolic pathways or catabolic pathways. Those roots in anabolic and catabolic tell us a lot about what that pathway is doing. Ana means up. So an anabolic pathway is actually assembling small molecules into larger molecules. So an example of this would be amino acids through an anabolic pathway will become proteins. Now, anabolic pathways, because you're building something, do require the input of energy. Catabolic pathways are the opposite. They're breaking down large molecules into smaller molecules. Cata means down. So an example of a catabolic pathway would be a carbohydrate breaking down, which is an energy-yielding nutrient, breaking down into carbon dioxide, which is an energy-poor product. So anabolic pathways require the input of energy. They're building something up. Catabolic pathways are breaking something down and releasing energy. Okay, so energy is the ability to do work. However, there are several different types, different forms of energy. The first type of energy I want to talk about is potential energy. Potential energy is quite simple. It is the potential for a system to do work. So when I hold my pencil up here, this has potential energy. Kinetic energy is associated then with movement. So when I drop my pencil, it transfers from potential energy here, and once it's moving, it's transferred down to kinetic energy. It's associated with movement. Next is chemical energy. Chemical energy is the type of potential energy that exists within chemical bonds. So when those bonds are broken, it releases energy. This is how living cells receive energy from food. Next is activation energy. Activation energy is the amount of energy needed for a reaction to occur. We'll talk more about this later, but enzymes actually can lower that activation energy, so it lowers the amount of energy needed in order for a reaction to occur, therefore speeding it up. Lastly is free energy. So in bio and AP Bio, you're going to hear it as Gibbs free energy. Gibbs is it's named after the scientist that discovered it, who developed the measurement for it, I guess. It is the amount of usable energy in a system. So I'm going to put the formula for calculating Gibbs free energy up here in a second. So take some time to really look at that formula and at the graph that I'm going to put up there, so that way you can see why calculating Gibbs free energy is important in relation to spontaneous or not spontaneous reactions. All right, as you are looking at that formula for calculating Gibbs free energy, note that the delta G represents free energy. Delta H is the enthalpy change, which is just the difference in bond energies between products and reactants, also known as heat. T is temperature in Kelvin. Delta S is then the entropy change, which is a measure of the disorder in the system. Now, looking at the graphs, we should see a couple new terms on here, exergonic and endergonic. First of all, exergonic is just saying that free energy is released from that reaction. So it is exiting the system, looking once again at those roots like exocytosis, exit, exergonic, releasing heat. Now, on the other hand, we have endergonic, which means that energy is added to that reaction in order for it to occur. These things really they make sense when you actually look at the graph. So on the y-axis we have Gibbs free energy, and we'll note that on the left-hand side, the starting point is much higher than it is on the graph on the right-hand side. And then we have a flipped reaction. So the exergonic reaction is spontaneous. Spontaneous, by definition, means occurring without any external prompting. So that reaction is happening on its own without the need for energy to be put into that system, which makes sense. On the other hand, we have endergonic reactions. That reaction is not going to be spontaneous because it requires energy to be added into it. So as you can see, by calculating the delta G, or the Gibbs free energy, it shows us a lot about that reaction, whether it's going to release energy, take in energy, and be spontaneous or not spontaneous. All right, the next concept I want to talk about as far as energy specifically are the laws of thermodynamics. For AP Bio, we are focusing in on the first two laws. The first law of thermodynamics states that energy is neither created nor destroyed, it is conserved. So it's changing its form. The second law of thermodynamics states that when there is an energy transfer, it's not always perfect. There will be some loss of energy into an unusable form. So remember that energy isn't just disappearing, it's not gone based off of our first law of thermodynamics, but it's transferring into something that you cannot actually use, such as heat energy. And when that does occur, it is actually increasing the disorder of a system. We hear a ton about ATP and how it is the energy currency of the cell, but I want to break that down into what that actually means and how it's relevant. So ATP is a small, relatively simple molecule that harnesses energy within its bonds. So every ATP molecule is made up of an adenine, a ribose, and 3 phosphate groups. And it has that potential, so potential energy, to break those bonds, the chemical energy, and release a quick burst of energy for cellular work to be done. What happens when those bonds are broken is it's actually the 3rd phosphate group that is broken off to create ADP and then a phosphate group, and that process alone releases free energy, which allows the system to do work. Now, what does that have to do with reaction coupling? Reaction coupling is when an energetically favorable reaction, so an exergonic reaction, like ATP hydrolysis, is linked with an energetically unfavorable reaction, so an endergonic reaction. This often happens through a shared intermediate, so the product of one reaction is then picked up and used as a reactant in the second reaction. So this is, once again, this is reaction coupling. It's really just taking advantage of the products and reactants available. One example of this is oxidative phosphorylation, which is the final step in cellular respiration. And I'm going to show you that picture now. Here we have a great picture of oxidative phosphorylation. So I want you to notice that the portion on the left is the ETC, the electron transport chain, and on the right is chemiosmosis. So what's happening is that in the electron transport chain, electrons are being passed from one molecule to another, and energy released in these electron transfers is used to form an electrochemical gradient. And then in chemiosmosis, the energy stored in the gradient is then used to make ATP. So the electron transport chain does need ATP synthesis, and ATP synthesis needs the electron transport chain. We are on to our last portion of this particular video. Thank you for sticking with me. This last part talks about enzymes, so I'm going to flip it over to a picture and voice recording so that way I can show you the different types of enzymes. The first picture you're looking at here is the induced fit model. So that's when slight changes are occurring at the activation site. So you can see that the substrate is entering the active site of the enzyme. Small changes in the shape of the enzyme then occur, and then the substrate is converted into its product and then released. Now, enzymes, their purpose is to lower activation energy. However, we can get inhibitors that decrease enzyme activity. This A here is the normal binding, normal enzyme function. Then we have competitive inhibition. What happens here is that other molecule is fighting for the activation site, not allowing the substrate to enter the activation site. We also have non-competitive inhibition, which enters at a different site than the activation site. However, it still renders that enzyme ineffective. The last picture we have is allosteric inhibition and activation. So when it's inhibition, the inhibitor enters the allosteric site and then changes the shape of the enzyme, not allowing the substrate to enter the activation site. In allosteric activation, we have a different process in which the activator enters that site and alters the shape of an enzyme for a positive way to allow that substrate to enter the active site. I hope that this video helped you girls understand energy and metabolism a little bit better. If you still have questions, I would recommend looking at Chapter 8 of your textbook. That's where most of this information is from. Also, I have the presentation and a reading guide, more videos, and other resources below. Up next, we will be talking about photosynthesis.